All right, hello everyone. Thank you very much to the organizers of this conference for the opportunity to present some carrot research. This is a very nice place to come to visit to give some scientific expertise. So I'll be talking, yes, about where we're going in the future with carotenoid studies. I work in the same lab with Dr. Phil Simon and Massimo Arizzo, so this will be a presentation showing how our lab is utilizing the carrot genome moving forward. So, presentation overview. I understand that the talk after me will give you a very in-depth overview of carotenoids and carotenoid accumulation, so I'll kind of breeze over this and just touch on what carotenoids are and our current understanding of accumulation mechanisms in carrot. Then the main focus of the talk will be on utilizing the technique of genotyping by sequencing to identify novel marker trait associations at the whole genome level. And then I'll show how we've used this technique in our lab to focus on several different carotenoid traits of interest, including lutein accumulation and beta-carotene and lycopene accumulation, and then kind of where we are right now and future directions for this work. So why do we study carotenoids? Well, they play an essential role for plant life through photoprotection, light collection. They also lead to the biosynthesis of abscisic acid and strigolactones. And they're also very important for animal life, or mammalian life, such as us, because they are the precursors for vitamin A, so it's very critical for our vision, as well as some of the other carotenoids have been shown to be involved with having anti-cancer effects, maintaining a healthy immune system, and reducing the risk of heart disease. Carotenoids can also be used as food colorants in cosmetics or in some pharmaceuticals. So, what are the mechanisms that plants use to accumulate carotenoids? I'll talk about these three mechanisms. So on the right, you can see the carotenoid biosynthetic pathway with the three compounds that I'll be looking at in this presentation, highlighted in their respective pigment colors. So there can be accumulation through the transcriptional regulation of the genes controlling carotenoid biosynthesis, so what's going into the pathway. If there's more that goes into the pathway, you accumulate more. There's also transcriptional regulation of the degradation of these carotenoid compounds, so if enzymes in the pathway are blocked or mutated, then you can accumulate that compound directly above it. But then a third mechanism for accumulation is the storage of carotenoids, or the formation of chromoplasts or transport of these molecules in the cell. And that is something that we have not focused on very much in carrot, but that's storing the carotenoids in carotenoid sinks. So what I'd like to point out here is, although we have a very good understanding, or a good understanding of the carotenoid biosynthetic pathway in carrot, that there are some genes that might not be involved in this pathway that are important to include in our carotenoid accumulation studies for possible candidates. So I'm not going to talk about this slide. There's a lot. This is a lot of the work that has been done in carrot looking at carotenoid biosynthesis genes and accumulation mechanisms. So there's been a lot of identification of homologs of these genes using other plant systems, and I think that the next talk will focus on a lot of these studies as well. But while there's been a lot of work done, there's still a long way to go. There's still very few links between actual genes and the traits that they control. So this is why I think that there's a need to take advantage of some of the novel technologies to explore carotenoid accumulation, not only at the biosynthetic gene level, but at the whole genome level. And to do that, I propose that we use genotyping by sequencing. I'll refer to this as GBS for the rest of the talk. So I'm going to demonstrate the utility of genotype GBS through three trait examples: lutein, lycopene, and beta-carotene accumulation. So this is a very brief workflow of what GBS all involves. In this case, I've been using mapping populations. So you extract DNA from these populations, you prepare your GBS libraries, which are then submitted for sequencing using next-generation sequencing technologies. You run the data through your sequence that you get back through a bioinformatic pipeline that identifies SNPs, and then you use this SNP and use mixed linear models to include the phenotypic data, as well as population structure and the SNPs to hopefully identify regions of interest. So I'll go through each of these steps a little bit more slowly. So the GBS library prep, here you can see a 96-well plate that represents where all of your DNA individuals from the population would be plated, along with a barcode or an adapter. So that's the way that you are able to separate all of your samples, by adding your DNA with an adapter, digesting the DNA, and then ligating that adapter or barcode to your DNA. So then you're able to pool those 96 samples together and still later through bioinformatic analysis pull them apart. After this step, you amplify these fragments, and then you can use a size selection for getting even distribution throughout the genome, and those libraries pooled together are then what you submit for sequencing. In our lab, we've used initially the Illumina HiSeq 2000 for sequencing, and now we're using the Illumina HiSeq 2500. A few things to consider with the library prep. You need thorough digestion of your DNA, because if you're only partially digesting your genome, you will only get partial representation across your genome. You also, if you have parents or very important samples in your population, because the coverage across genotypes after pooling isn't necessarily perfect as much as we'd like it to be, you might want to include these very important samples in replicate or in triplicate. And finally, coverage is important. Carrot is a relatively small genome, so that's nice. GBS works very well with carrot. It is highly heterozygous, though, so you do want to consider that for calling heterozygous SNPs, that you have appropriate levels of coverage. So I'm not going to talk very much on the bioinformatics pipeline. So I use something called the Tassel pipeline, which comes out of Ed Buckler's group at the University of Cornell in New York in the United States. And ultimately, we've been able to utilize the carrot genome, the reference, and all of these reads are demultiplexed after we get the sequence back. So they're broken into their respective barcodes, aligned to the genome, and then SNPs are called based on the alignment to the genome. And these SNPs can be filtered for coverage or for allele frequency or inbreeding coefficients. So more information, please come see me if you want to talk about the bioinformatics, but all of this software is freely available at mazegenetics.net/tassel. So there's also a graphical user interface, or GUI, that you can look at your data. So this is very useful for looking at alignments of your final SNPs. You can also create phylogenetic trees, look at linkage disequilibrium, and very importantly, with the Manhattan plots, you can look for associations throughout the genome of genomic location with significance of any given trait that you're phenotyping. So there are some strengths and some weaknesses with this GBS pipeline. One of the strengths is just the sheer number of markers you get from the pipeline. You can get upwards of over 100,000 markers in a single 96-well plate. These markers don't have an ascertainment bias because they're developed with the experiment. You're not using something like an array where SNPs were developed on a previous panel of genotypes. It's relatively low cost. So for a single GBS lane at Wisconsin, we typically spend somewhere between $2,500 and $3,500. So that's with the sequencing facility that we have. And all of the analysis tools are freely available. So the major weakness of the pipeline is that there can be a problem with missing data. Sometimes the genotype can drop out because it wasn't barcoded correctly. But what we've realized is that the missing data isn't such an issue because normally you're limited by the number of genotypes, not by the number of markers. So this is not such a big deal. It does cost some money, so that's always a drawback. It is a low cost per data point, but can still be a little bit of a large upfront cost for the lab. If you are not familiar with bioinformatics, it can be a little bit difficult to start that, but I did not have much training, and I have done it, so you can too. And then finally, genome availability. You'll soon have a carrot genome to do this on, so don't worry about it. All right, so Tassel GBS pipeline in carrot. So I've used 3 various mapping populations, 2569, which is a yellow versus orange population segregating for beta-carotene, 1061, which is a red versus orange population segregating for lycopene accumulation, and 5249, which is a white versus yellow population segregating for lutein accumulation. I'd like to point out that the last 2 experiments, we only used a single GBS plate. So this is to demonstrate, if you're using a relatively small number of samples, how much bang for your buck you can get. That's not going to translate well. I don't know. Okay. So for the phenotyping, we used high-performance liquid chromatography as well as a 0-to-1 visual assessment of pigment. So these are the typical HPLC chromatograms for white, yellow, red, and orange. And just to remind you, so DNA was extracted from these samples, the libraries were prepared, we called the SNPs, and then we use Tassel to identify regions of the genome that are associated with each of these traits. So to get right into the results, our first population segregating for beta-carotene. If you look at the top left corner there, this trait segregates as a simple Mendelian trait for 3 yellow to 1 orange. And using GBS, we were able to identify over 31,000 SNPs after filtering for 5% missing data for marker, 10% missing data for genotype. And what you can see here is the Manhattan plot. On the y-axis is the negative log of the p-value. On the x-axis are the 9 carotte chromosomes. So you see a very significant peak situated on chromosome 7. You can then go in and look at the actual genotypic calls of that region. And what you see is the recombinants in that population flank a region of 275 kb. For the lycopene results, we realized that this population was segregating 15 to 1, non-red to red. So it was under a 2-gene model. We identified 25,000 SNPs through the GBS pipeline with the same level of missing data. And we were able to identify 2 regions, one on chromosome 1 that was over 7.5 megabases, and another region on chromosome 3. So again, you go in and you can find those actual recombinants using your markers to find where the borders of those events are. So important to note that this was carried out in only 88 samples. And the final population for lutein accumulation, so white versus yellow, again another nice Mendelian trait, 3 to 1. A fewer number of SNPs, 23,650. And we found a very significant region on chromosome 5 that is less than 200 kb, so a very, very well-defined region of the genome. So the work that we're doing now is we're adding functional or expression assays to this work. So we have conducted an RNA sequencing experiment using orange, yellow, and white genotypes. So this is specific for the first 2 mapping populations for beta-carotene accumulation and lutein accumulation. And with these very narrowly defined regions, once you layer the differential expression data on top of it, we're very happy to see that there are only 2 differentially expressed genes within the region of interest for beta-carotene and 1 differentially expressed gene in the region for lutein accumulation. So those are very strong candidates. This expression work is being verified with quantitative PCR. Fine mapping is being conducted in the region of interest using a genome-wide association panel. So we're using some materials from the plant introduction collection to further narrow this region, as well as advanced mapping populations. And then we are in the process of initiating the functional verification of our candidates, both through rescue experiments or transformation and knockdown experiments with RNAi, or perhaps looking into the CRISPR-Cas9 system. And interesting for an applied focus is these markers then can automatically be converted into useful codominant markers that you can use in the breeding program, which will be used for marker-assisted selection. Oh, and I'm not talking about this. We're also using GBS in some of the other Apiaceae. I'll be giving a poster on this, so I'll talk about that tomorrow. But it can apply to all different types of crops. So with that, thank you very much, or merci beaucoup. That's me at age 3, standing in some Apiaceae. So maybe you know what your career is very early on. And yeah, thanks to Phil and Massimo very much for all of their work. Doug Sinalik does the bioinformatics, Megan Bowman for the RNA work, as well as Rob Kane for field assistance, and special thanks for NSF, USDA, Madison, and the Carrot Genome Consortium.
I'm sorry, I think I have a very quantitative question. You said that the cost of GBS is around $3,000, and I'm not sure I've well understood, because at first it sounded like it was this price per genome, and then it sounded like it was per population, but I think it's per genome, right?
So that number is on a per 96 well plate, or a single Illumina sequencing lane. So however many samples you put on your lane that you barcode together, could be 96, could be 384, it's that number. So more of a population than per genome.
Okay, thank you.
Yeah.
From Vilmarin. Maybe I missed information, but you didn't mention the sequencing effort that you would recommend in carotte for performing GBS, especially considering the heterozygosity.
So sequencing method being the sequencing platform?
The depth of sequencing. How many X you would recommend to be able to have a good data set? So
I did not suggest it because we have not used so much of a filtering based on depth as we do for missing data. Along with the GBS, you can also get a coverage, and typically we see somewhere between 8 and 10x as being a useful coverage for being confident with calling a SNP. With the pipeline, sometimes we see that there are more heterozygous calls, like maybe there are 25 reads and you have one allele and 24 of the other allele. So we do have a parameter that we filter out alleles that are not in the population that are less than 30% of the heterozygous call. So more so than the coverage is this other parameter to get rid of excess heterozygosity. But 8 to 10x, simple answer.
May I have a second question? You were talking about conversion of the markers in codominant markers. Did you use the reference genome for doing it, or could you do it without any reference, like a basic Casper assay? So
I used the reference genome, but one thing that you could do, so the GBS it's a sequence read, it's 100 base pair read, and the pipeline trims it to 64 base pairs, and that's where the SNP is identified. So you could certainly begin to look at 2 nearby SNPs and try to develop primers within those 2 reads, and then those 2 reads could amplify something that you could begin to develop a primer without the genome. Well, yeah, so you would have to use linkage disequilibrium to look at markers that are close to each other, but it can be done. Or just use the 100 base pair reads and have a very small amplicon.
With a SNP in the middle, because if it's on the edge, it's impossible.
Yes, but SNPs are, I mean, quite common, so at the rate where I am, you would just have a significant SNP within 100 kb, you can find one with a SNP in the middle.
Okay, John.
Hi. You started with the mapping population, which segregated, which is very nice. Imagine you do this in the wild population. What do you think the coverage should be in sample size to try to get an answer?
Yeah, so I mean, in a way, using an association panel can be better for narrowing down the region of interest, because the mapping population, you get a lot of linkage drag with the region of interest. So you definitely have to increase your coverage, but more so than that, you just want to make sure that your allele frequency is high enough in the population you're sampling to be able to capture it. So if you're filtering on less than 5% allele frequency for some very uncommon trait, then it will be very hard to detect. So maybe like this sampling strategy to try to include the trait of interest, so if it's a phenotypic trait, to make sure it's more along the lines of 50% or just above 5%. But in one of the techniques that you can use for this is, along with the genotyping by sequencing, doing very low coverage resequencing to use the low covered resequencing as something you can use imputation. So it fills in all of the missing data, and it also confirms where you believe the recombinations are. So I don't have an actual answer for a coverage that I would suggest, just techniques that you use to use in an association panel, which is what they do all the time in maize and some of the big crops.